Students, welcome back. Today we are just going to discuss the topic gametogenesis. Till now we have completed the complete structure of the male reproductive system, female reproductive system, and the regulation of male and female reproductive system, how the hormones are involved in the regulation of male and female reproductive system. Now today we are just going to see the process of gametogenesis. This is the first physiological process or first reproductive physiological process. So here we are just going to start with the process that is called as gametogenesis. Gametogenesis means to say the process where gameto plus genesis. Gameto means gamete, genesis means formation. So the process in which the gametes are being synthesized or formed are called as gametogenesis. So here directly we are just going to say the formation of gamete is known as gametogenesis. You know very well what is the meaning of gamete. Gametes are the specified sex cells produced by the male and female. Male sex cell and the female sex cells, they are called as the gametes. These sex cells are being provided by each of the parent during the sexual reproduction to continue the process of reproduction, the sexual reproduction. So here the gametes are the sperm in case of the males and the egg in case of the female. So the formation of egg and sperm is called as gametogenesis. And you know very well from the earlier time only we are discussing it. The process of gametogenesis is taking place or completing there inside the primary gonad. What is the meaning of primary gonad? We know very well. The primary gonads are defined on the basis of that the structures which are involved in the formation of the gametes. So here the primary gonads are testis for the formation of the sperm. In case of the male, the sperm is the male gamete. So the testis is responsible for the formation of the sperm. And in case of the female, the ovary is responsible structure for the formation of the egg. And remember, in the previous lecture only when we were discussing about the hormonal regulation or hormonal control of the male and female reproductive system, that time we have seen the complete process is being regulated by the mean of some hormone. Those hormones are called as gonadotropins. These gonadotropins are being released from the pituitary gland. So these gonadotropin, if you have remember, FSH, LH and ICSH. GnRH was released from the hypothalamus to regulate the release of these hormones from the pituitary gland. And one more important thing that I'm just going to say I'm going to discuss with you, the vitamin E. Vitamin E is called as anti-sterility vitamin. Anti-sterility vitamin means to say the vitamin which prevents the sterility. You know very well, yes, in the previous lecture only we have seen the sterility means what? The male or female is not having capacity to give to the young ones, or male is not having capacity to provide the enough amount of the sperms thereon, or the male is not having ability to produce the healthy sperms thereon. So vitamin E is anti-sterility vitamin, means this is required for maintenance of the proper reproductive functions. This is essential for the gametogenesis. This is essential for the gametogenesis. Remember, on the basis of the gametes formed, the gametogenesis is of two types. Gamete form means what? We have two types of the gametes. One is called as the male gamete, another one is called as the female gamete. So here two types of gametes are there. One is called as sperm. So formation of sperm is called as spermatogenesis. The process will occur there inside the testis. The formation of sperm is called as spermatogenesis, and the process is completed there inside the testis. And similarly, the formation of egg is called as oogenesis. The process will get complete there inside the ovary only. So spermatogenesis in testis, formation of the sperm. Oogenesis, the formation of egg inside the ovary only. And we are just going to discuss one by one the process. Now we are just starting the process that is called as spermatogenesis. Once again we are just starting with the process spermatogenesis. You can understand well what is the meaning of spermatogenesis. Spermato plus genesis. The word spermatogenesis means what? Spermato means spermatozoa, genesis means formation. So the formation of male gamete, that means sperm. The formation of male gamete, that is called as sperm, is called as spermatogenesis. You knows very well from the previous lectures only, again and again we are discussing that the formation of male gamete or spermatozoa is getting completed there in the testis, and the actual functional unit and the structural unit of testis is called as seminiferous tubule. So actually the formation of male gamete or sperm is being taking place there inside the seminiferous tubule of the testis. So the site of spermatogenesis is the seminiferous tubule of the testis. So now we are just going to understand the mechanism, how these sperms are being produced inside the seminiferous tubule, how these are being produced inside the seminiferous tubule. So how the process is getting completed. So to understand the process, to understand the easiness of the process, the two types of the gametes are there. two steps are there to involve in the formation of spermatozoa. The first step is called as formation of spermatid, and second step is called as spermiogenesis or spermatogenesis. Are you getting my point? The spermatogenesis process completes into two steps. One is called as formation of spermatid, another one is called as spermiogenesis or spermatogenesis. So now moving on to the first step, that is called as formation of spermatid. So formation of spermatid. So we are just moving to discuss the formation of spermatid. So the procedure will start from the primordial germ cell. If you have remember, during the structure of the seminiferous tubule, during the structure of TS of the seminiferous tubule, we have studied about that the primordial germ cell is going to divide mitotically and going to produce some of the cells called as spermatogonias, primary spermatocyte, secondary spermatocyte, everything. So the process of formation of spermatid is begin from the primordial germ cell of the seminiferous tubule. Primordial germ cell of the seminiferous tubule will start the process of spermatid formation, and here remember the process of spermatid involves the division. What type of divisions are involved there in the formation of spermatid? In the process of formation of spermatid, both mitosis as well as the meiosis. Are you getting my point? So the process of spermatid formation is begin from the primordial germ cell, and here to form the spermatid from primordial germ cell, the divisions are required. Those divisions are called as the mitosis and meiosis. Now we are just going to see in detail how the process is being completed here on. Here the process of spermatid formation completes into the three steps process, or we can say that it is a three-step process. The three steps are multiplication phase, growth phase, and the maturation phase. One by one we are just going to see the phases. One by one we are just going to understand the process. The first phase is called as the multiplication phase. Multiplication phase means to say the phase of multiplication of the cells. When the cells are getting multiplied from the previous number of the cell, here number of cells are getting increased many of the time, increased in manifold of the time. So here what happened, the growth in the number of cell is being reported, the growth of number of the cell is recorded. Here what happened, the process of the mitotic division begins in the primordial germ cell. The primordial germ cell will enter in the process of mitotic division and will continuously divide mitotically and will form two types of the cell. One cell is called as spermatogonia A and spermatogonia B. Basically the cells are the common cells, those are called as spermatogonia, but based on their functional behavior, the cells may be referred as spermatogonia A and spermatogonia B. Remember here the primordial germ cell was diploid cell and it is going to form mitotically the diploid cell, that is called as spermatogonia. So spermatogonia is also diploid and primordial germ cell is also diploid. So during multiplication phase what happened, the complete mitotic division is there, and due to the mitotic division multiplication in the number of the cell is being occurred or observed, and here what happened you can understand well. The primordial germ cell undergoes in the process of mitotic division, and due to the mitotic division the primordial germ cell give rise a great number of the new cell. Those cells are called as spermatogonia, and based on their function the spermatogonia may be divided on two different groups, the spermatogonia A and spermatogonia B. What are these spermatogonia A and spermatogonia B? Remember, some of the spermatogonias are going to divide continuously to give rise the new spermatogonia. Are you getting my point? Out of these spermatogonia, out of many of the spermatogonia produced, some spermatogonias are going to divide again and again to produce the new spermatogonia. That type of spermatogonia are called as spermatogonia A. Are you getting my point? Spermatogonia A, the continuously divide to form the new spermatogonia. What are spermatogonia A? These are the spermatogonia which are continuously divide to form the new spermatogonia. And what are B? Any one of those spermatogonia produce new one. Any one of the spermatogonia is undergone is the process of enlargement and forms a new cell that is called as primary spermatocyte. So are you getting my point? Some of the spermatogonia undergoes in the continuous mitotic division to produce the new spermatogonia called as spermatogonia A, and some of them are undergoing in the process of enlargement of themselves, and that is called that is going to form the primary spermatocyte. So A will continuously divide to form the new spermatogonia, while the B will form the primary spermatocyte. So here the first phase after the formation of primary spermatocyte, or after the formation of spermatogonia, the first phase, that is multiplication division, is getting completed. Now this spermatogonia B enters in the process of the enlargement, that is called as growth phase. So first one is multiplication phase, where great number of the new cells produced, that is called as spermatogonia. Now this spermatogonia undergoes in the process of growth. So growth phase is there. What is there in growth phase? During the growth phase, the size of any one of the spermatogonia increase in the size, enlarge in the size, and appears as a different cell or distinct cell from the other spermatogonia. That spermatogonia is referred as the primary spermatocyte. Are you getting my point? What happens during the growth phase? The spermatogonia, any of the spermatogonia, enlarge in the size and appears distinct from the other spermatogonia, that is called as primary spermatocyte. How this enlargement occurs? So at the time of enlargement, the spermatogonia, since the any one of the spermatogonia has to undergo in the process of maturation division, will have to continue the maturation in form of the sperm, so it is required to accumulate the nuclear material as well as the cytoplasmic material. What it does, the spermatogonia, which has to undergo in the metamorphosis as the spermatocyte, so that will increase in size, enlarge in size due to the accumulation of the nuclear material and the cytoplasmic content. The enlarged spermatogonia is called as primary spermatocyte. Remember that primary spermatocyte is also a diploid cell. So here till now we can understand the primordial germ cell diploid, spermatogonia diploid, and here no division involved. Remember, in the growth phase there is no division involved, only the accumulation of the material is involved. So due to the accumulation of involved, new cell is formed, so that is also the diploid cell, no division. And remember one important point, we'll discuss the differences between oogenesis and the spermatogenesis. That time this will be an important difference. The growth phase is going to mark a great difference between oogenesis and the spermatogenesis. The oogenesis growth phase is very large phase, or longer phase, and here the growth phase is very much shorter. So is it clear now? In multiplication phase, multiplication; in growth phase, enlargement; and primary spermatocyte, that is diploid, is being produced. Now this primary spermatocyte undergoes in the process of maturation division or maturation phase. So the process of conversion of primary spermatocyte in form of the spermatid is called as maturation phase. During the maturation phase, what happened? The primary spermatocyte, the diploid primary spermatocyte, is going to convert in form of the haploid spermatids. So what happens if a diploid cell is going to convert in form of the haploid cell? So definitely which kind of division is there? Meiotic division. Remember my dear child, the spermatid formation is the process where we have reported what type of division: mitotic division and meiotic division. The complete process of multiplication division involves the mitotic division, while the maturation division or maturation phase will have the meiotic division. So what happens? The primary spermatocyte, which is received after the growth phase, enters into the maturation phase. Enters into the maturation phase means what? That primary spermatocyte will undergo in the process of meiosis, and you know very well the meiosis is a sequential process to subsequent meiosis is there, that is meiosis first and meiosis second. So primary spermatocyte is undergone in the process of meiosis first and going to form the secondary spermatocyte. Are you getting my point? Primary spermatocyte is undergoing in the process of meiosis first and going to form the secondary spermatocyte. The secondary spermatocyte is undergone in the process of second meiosis and forms the spermatid. So remember, the primary spermatocyte is the diploid cell, will undergo in the process of meiosis division first and will form a haploid cell, that is called as secondary spermatocyte. Now this secondary spermatocyte will undergo in the second meiotic division and will form the spermatid. So from one primary spermatocyte, two secondary spermatocyte, and now two secondary spermatocyte will enter in the meiosis second, so will produce four spermatid. So that is why we can say one primary spermatocyte is equal to four spermatid. Means what? One primary spermatocyte will get converted in form of the four spermatid. So once again I am just going to repeat the complete process. Multiplication phase, The mitotic phase where new cells are being produced, that is called as spermatogonia. Growth phase, any one of the spermatogonia enters in the growth phase and enlarge in the size to form the primary spermatocyte. Maturation phase, that primary spermatocyte undergoes in the meiotic division and form the haploid spermatids. So one primary spermatocyte will give rise to 4 spermatids, remember here. And the formation of spermatid occurs at the place here only, because now they are not having enough amount of energy or nutrition so that they could convert themselves in form of the functional sperm. So they are leaving the place of the primordial germ cell or the germinal epithelium and moving on to the another cell present on the germinal epithelium or in the seminiferous tubule, that is called as the Sertoli cells. So spermatid, after completion of their division here, so the mature spermatid or the spermatids whatever is being formed, they are moving on to the Sertoli cells and getting converted in form of the sperms. What they does, they are just leave out the place of the formation and moving out to the Sertoli cells, getting attached to the Sertoli cell for the further development in form of the sperm. So is it clear to you all? Remember, the spermatid will get converted in form of sperm on the Sertoli cell. So is it clear to you all? Here, if we could see the process, a diploid primordial germ cell was there, the PGC. The 2N means the diploid cell has undergone the process of mitosis, forms the diploid cells. Once again they are just undergoing in the repeated division, again and again the division will be there, and all these new cells are called as spermatogonia. The two types of spermatogonias are there. These spermatogonias are called as spermatogonia A, which continues the division to form the new spermatogonia. Any one of the spermatogonia will undergo in the process of enlargement due to the accumulation of nuclear and cytoplasmic material and will form a large cell that is called as primary spermatocyte. Now this primary spermatocyte will undergo in the process of meiosis division first, reduction division first, or the maturation division first. Due to the meiosis, the number of chromosome gets half, and N means what? This is the haploid cell. Till now all the cells were diploid. Now the haploid cell is produced. That haploid cell is called as secondary spermatocyte. This secondary spermatocyte once again moves in the process of meiosis division second, reduction division second, maturation division second. Now due to the meiotic division, each of secondary spermatocytes are going to form the new cells that are called as spermatids. So is it clear to you all? Once again I am just going to say, the primordial germ cell undergoes in the process of repeated mitosis, forms the diploid cells that are called as spermatogonia. Now the spermatogonia enlarges in the size to form the primary spermatocyte. Meiosis first, meiosis second. The primary spermatocyte will form the two secondary spermatocyte from the meiosis first. Then secondary spermatocyte will move into the process of meiosis second to form the 4 haploid spermatids thereon. Remember the process of formation of spermatogonia. So this phase is called as multiplication phase because the new number of cells are being increased again and again. The second one, where there is no division involved, only the enlargement of the cell enlarge, that is called as the growth phase, where spermatogonias are getting converted in form of the primary spermatocyte. The third one, where the primary spermatocyte is getting mature in form of the spermatid, that is called as maturation phase. That phase is dedicated for the meiotic division only. So this is what a flow chart of the formation of spermatid. Now we are just going to understand how this spermatid are getting converted in form of the mature sperm. So just take a look. So students, after completion of the process of spermatid formation, the second step of spermatogenesis begin, that is called as spermiogenesis or spermatogenesis. Spermiogenesis is nothing, it is only the transformation of spermatid into the sperm. So that is occurs onto the Sertoli cell. So metamorphosis of spermatid on Sertoli cell is called as spermiogenesis. Actually what happens during the spermiogenesis is the conversion of spermatid into the sperm. What happens during the spermiogenesis is the spermatid is going to convert in form of sperm. How that is being going on. So here what happened, there is no division involved. Remember, The spermiogenesis is not the process where division is required, so there is no division. Only the differentiation of the spermatid is going to lead to develop a structure that is called the sperm. So what happens? No division involved, only the division of only the differentiation of spermatid is there. What kind of differentiation, what kind of the changes are being observed during the differentiation of spermatid into the sperm? Remember, till now the spermatid is considered as the heavy structure and the non-motile structure. Till now the spermatid is heavy and non-motile structure. During the differentiation, during its transformation, during its metamorphosis in form of sperm, it has to reduce its weight and has to develop the capacity of movement. Are you getting my point? Normally the spermatid is non-motile and heavy in the weight, but during the time of or during the process of metamorphosis, it has to reduce its weight and to develop the capacity of movement. Why this is required? Because the movement of sperm is extremely important in the female reproductive tract, because egg is non-movable structure, non-motile structure. The sperm has to cover the complete journey of the female reproductive tract so that it could reach to the site of fertilization. So what is the objective of formation of sperm from spermatid? Reduce the weight and convert it in form of the motile structure. When it is going to complete its develop, so the complete process occurs there on the Sertoli cell. The complete metamorphosis occurs on the Sertoli cells, we know as well. If you have remember, on that day we have just discussed the diagram. The spermatids are found to be attached on the cusp of the Sertoli cells. Here they are just getting the nutrition, everything for the purpose of conversion itself in form of the functional sperm. So here they are deriving the nutrition, they are getting protected from the harsh environment. So here they are getting protected, they are getting nutrition, and when they are just getting converted in form of the fully mature sperm, fully metamorphosed sperm, so they are just going to release out from the Sertoli cells and moved into the lumen of seminiferous tubule. So releasing out of the sperm from the Sertoli cell and moving into the lumen of seminiferous tubule is called as spermiation. What is spermiation? Spermiation is the release of mature sperm in the lumen of seminiferous tubule. What is spermiation? After the formation of complete mature sperm on the surface of Sertoli cell, they are being released into the lumen of seminiferous tubule. Okay, so now we are just going to observe what are the changes, what are the processes are being observed during the spermiogenesis. What happens? These are very important points that I am just going to discuss. The changes are there. The nucleus of spermatid is going to form the head of the sperm. Remember what happened, what are the changes? The nucleus of spermatid is going to form the head of sperm. The Golgi body of the spermatid is going to form the acrosome. We'll discuss in the structure of the sperm. Golgi body of the spermatid is going to form the acrosome. Acrosome is very important in the structure of the sperm, and it is being covered externally by the acrosomal membrane that is also being formed by the Golgi. Rest of the Golgi body are going to form the acrosomal cap. Now the centriole is going to form the proximal and distal centriole. Along with that, the distal centriole is going to form the tail and middle piece also. Mitochondria is a great source of energy and form the middle piece. Are you getting my point? Middle piece is there, so middle piece will be formed by the mitochondria so that it could provide the energy for the movement of the sperm. So now let us take a look on the structure of the sperm. If we are just going to discuss the structure of the sperm is divided into 4 different regions. The structure of the sperm is divided into 4 different regions. The first region is called as head, second one is called as neck, third one is called as middle piece, and fourth one is called as tail. So what are these structures? What are these regions? The head, neck, middle piece, and the tail. One by one we are just going to discuss all these structures. The head means the topmost region of the sperm. The head of sperm is being produced by the nucleus of spermatid. Nucleus of spermatid is going to form the head of sperm. And remember, the head of sperm will have the genetic material, since it is being produced from the nucleus, so that will have the genetic material. Basically the chromatin network is present, and the chromatin network is mostly having the DNA in present there, or DNA is present over there. So nucleus, head, nucleus is going to form the sperm head. So sperm head will have the genetic material, basically what, in form of the chromatin network, and they are in form of the DNA mostly. And sometime that has been reported they are having rich in some basic protein, the protein of the basic nature called as protamins. So remember what is there in the sperm head. The sperm head is completely made of the nucleus. So it will have the DNA or chromatin network and will have a basic protein that is called as protamin. That is the important question for the sperm structure. Protamins are present there in the sperm head and their basic nature protein. What happened? This is being reported. The sperm head is having a granular appearance over there. A granule is being present on the top of the sperm head. A sac is present on the top of the sperm head, that is called as acrosome. On the top of a sperm head, the acrosome, a granular structure is present, that is called as acrosome. What acrosome does? Acrosome is being produced from the Golgi body, and that is filled with the hydrolytic enzymes. Those enzymes are called as sperm lysine. Are you getting my point? The acrosome is being produced from the Golgi body and completely filled up with the hydrolytic enzyme. These enzymes are commonly called as the sperm lysine. We'll see their roles during the process of fertilization. We'll understand during the process of fertilization. But here you just remember these hydrolytic enzymes are very important during the penetration of the egg membrane at the time of fertilization. Egg is protected by variety of the membrane, many of the membrane. These membrane cannot be penetrated easily. So to penetrate those membrane, the sperm is already having the arrangement of the hydrolytic enzyme. These hydrolytic enzymes are making the sperm capable to penetrate inside the inside the egg body. Are you getting my point? Remember, but problem is that if that acrosome remains open, so that may hydrolyze any of the structure of the female reproductive tract, even though they are in the male reproductive system wherever the sperm is moving. So to protect from such kind of the disastrous nature of the acrosome is being covered externally by a membrane that is being also produced by rest of the Golgi membrane. Are you getting my point? The Golgi body is basically making what? Acrosome, but rest of the Golgi body is going to make a protective covering over the acrosome, that is called as acrosome cap or Gallia capitis, or we can say the acrosomal membrane, that is going to protect the acrosomal discharge. So that is being discharged. The membrane will get damaged only at the time of penetration into the egg cell. Is it clear? So now moving on to the next point. So after head, the next point is called as neck. Neck can be considered as a junction between the head and the middle piece. Neck is what? Neck is a kind of junction between head and middle piece. So it is present as in form of a narrow region. The head is going to be expressed, neck is going to express easily, can be understood easily, but this is only a kind of connection. Nothing special is present there only. The neck region is basically made of a kind of groove, and that groove is having the centriole located over there. So what is neck? Neck is a narrow part which connect the head and middle piece of the sperm. So moving on to the neck. So next point is the neck is having the centriole, and remember the centrioles are going to divide in two centrioles. The first centriole is called as proximal centriole and second one is called as the distal centriole. The proximal centriole is keep itself in groove present just below the sperm head, and distal centriole is lying just below the proximal centriole. Proximal centriole is also having very important function that we'll discuss later on. Just keep in your mind what is the function of proximal centriole. We'll discuss it later on. And the distal centriole is very important here in the structure of sperm. The distal centriole is working as a basal body. My dear student, you knows very well the basal body means the structure which is going to give rise any of the thread-like structure, flagellated structure, the cilia, flagella, anything. So it is working as the distal centriole is working as the basal body. So it gives rise a filamentous structure that is called as the axial filament. What is distal centriole? The second centriole which is lying just below the proximal centriole, that is going to work as the basal body. What it does, it is going to give rise a thread-like structure, a filamentous structure. That filament is called as axial filament. That axial filament runs completely from top to the bottom, means what, from the point of origin to the point of end of the structure of sperm, means from middle piece and tail it runs to complete middle piece and to the tail. What happens, just understand the common point, very important point I'm just going to discuss here. At the junction of middle piece and tail there is a ring present. What happens at the junction of middle piece and the tail of the sperm, ring structure is present. That ring structure is called as annulus or the ring centriole. Are you getting my point? At the junction of middle piece and the tail, at the bottommost region, at the junction of the middle piece, means at the bottom of the middle piece, at the junction of middle piece and the tail, ring structure is present, that is called as annulus, and that is also made of the centriole only, so that is called as ring centriole. Are you getting my point? At the junction of middle piece and the tail, annulus or ring centriole is present, a ring like structure is present, that is called as annulus or ring centriole, that is also known as the Jansen's ring. So is it clear? So at the junction of middle piece and the tail, the Jansen ring, ring centriole or the annulus is present, and the axial filament is passes in between these also, means what, it has to penetrate or it has to bisect these rings also. So these rings are present around that axial filament also. One more thing, in the middle piece only we have observed the presence of the mitochondria only. So when filament is moving in between the middle piece, so that time the mitochondria is going to arrange spirally on the axial filament only, and that is going to form the spiral sheath or the Nebenкern. Are you getting my point? In the middle piece, so what happens at the junction of the middle piece and the tail, the ring centriole is there, so it is going to bisect that, the axial filament is going to bisect that, but in the middle piece only the axial filament is having the spiral sheath of the mitochondria arranged over there, that is called as the Nebenкern. So Nebenкern is very important because the mitochondria is providing the energy for the movement of the sperm here only. So now we are moving on to the next structure. After neck, the next structure of the sperm is called as middle piece. What is middle piece? Middle piece, the part which is basically made of the mitochondria. It is known to provide the energy for the purpose of the sperm movement. What happens here, you have just seen, we have just discussed, the axial filament is being axial filament when moves in between the middle piece, the mitochondria are getting arranged on itself or on the axial filament, and they are just going to provide the energy. So the arrangement of mitochondria present on the axial filament is called as Nebenкern. So you can understand very well the Nebenкern is there. So due to the presence of Nebenкern, the Nebenкern is being arranged on the axial filament only. So that filament has to produce has require energy. So that energy is directly given to the filament only. So Nebenкern is there to provide the energy. And one more thing, so this middle piece is working as the upper cell. Around the Nebenкern, the cytoplasm is there. Outside the cell membrane is there. So the cytoplasm present in the middle piece region, or we can say the cytoplasm present outside of the Nebenкern, is called as manchete. Are you getting my point? The Nebenкern layer is covered externally with a thin layer of cytoplasm called as manchete. So what is manchete? The thin layer of cytoplasm around the middle piece. We can understand well, the manchete is a thin layer of the cytoplasm present around the Nebenкern. You can say the thin layer of cytoplasm which is present in the middle piece is called as manchete. Now moving on to the last part of the tail, that is called last part of the sperm, that is called as tail. The tail is the last part, the longest part of the sperm. Tail is helping in the movement of the sperm, locomotion of the sperm. Due to the tail, it is going to perform a specific waving movement, wriggling movement. The tail is also formed of the elongation of distal centriole. How the tail is being formed? From which part of the tail is being formed? So tail is formed of the elongation of the distal centriole. Remember, the tail is made of the protein fibers which controls the tail is made of the protein fiber which contract to give the characteristic movement of the sperm. So this is what the structure of sperm is there. We are just going to see the diagram of the sperm structure, or we are just going to see or observe the diagram of sperm. Here we are going to discuss the rough diagram of the sperm. Here what happened, the sperm head, the three part of the sperm structure is there. The entirmost part or interior most part is called as the head. This one is the head. the middle one is neck, this one is the middle piece, the third one, the last one is called as the tail. So this one is head, this one is middle piece, this one is head, this one is the neck, this one is middle piece, and this one is the tail. So 4 different parts are there. So just see the structure. Head is completely made of the spermatid nucleus. So if it is made of the nucleus, so it will have the genetic material, and genetic material is in form of the DNA, and DNA will have some specific basic protein that is called as protamines. So that is present in form of the chromatin network. We can say this is what a chromatin network, that is basically made of the DNA. At the top of the sperm head, what happened? Golgi body is going to form a specific granular structure that is having, or that is filled up with the hydrolytic enzyme called as sperm lysine, that is known as acrosome. At the top of acrosome, a protective covering is present that is called as acrosomal cap, acrosomal membrane, or gallia capitis. Just below the sperm head, the centriole is going to divide in 2 parts and arranged there inside the neck only. The proximal centriole, will discuss the role of proximal centriole later on. The distal centriole, we'll discuss. The distal centriole is working as the basal body, which gives rise the thread-like structure. That thread-like structure is called as axial filament. That axial filament is later on will convert in form of the flagella of the sperm. So axial filament is originated from the distal centriole only and moved in between middle piece, tail, and move outside in form of the flagella. So here we have seen in middle piece the mitochondria are getting arranged over the axial filament and form a specific structure called as Nebenкern. In middle piece, the axial filament will have the spiral sheath of the mitochondria that is called as Nebenкern, and that is required for providing the nutrition to the nutrition for energy for the movement of the sperm. Now when it moves downside, so at the junction of middle piece and the tail, annulus is present, a ring like structure is present, that is also made of the centriole, that is called as ring centriole or annulus or the Janssen ring. So that is also being present around the axial filament. Now the axial filament move inside the tail, and afterward that is going to convert in form of the flagella. Flagella is helping in the lacing movement and is formed of the proteins, contractile protein. That proteins are helping in the lacing or waving movement of the sperm. So here the last part of the body is tail. The longest portion of the sperm structure is tail. So here once again I'm just going to revise the structure of the sperm. What is the structure of the sperm? The sperm head, neck, middle piece, and the tail. The head is made of spermatid nucleus, will have the DNA and the chromatin network, will have the acrosome on the top, acrosomal cap is present there. The neck is having 2 centriole present there, proximal and distal. Distal centriole will work as the basal body and will give rise the axial filament. Axial filament will continue to arrange the mitochondria on the middle piece, that is called as Nebenкern. Then the ring centriole is present at the junction of middle piece and the tail. Axial filament will move into the tail and finally comes out in form of the flagella here on. Middle piece will have the arrangement of mitochondria. Along with the mitochondria, it is being circled by or is covered externally by the cytoplasm, that is called as manchete. So here the structure of spermatozoa is completed. Once again we are just going to revise the complete topic of the day. The complete topic of the day, we have just started with the gametogenesis. The gametogenesis is the process where the formation of gametes occur. The gamete formation occurs there in the testis and ovary. In testis, the male gamete is formed, that is called as spermatogenesis, and in ovary the female gamete is going to form, that is called as oogenesis. Today we have discussed the spermatogenesis. That spermatogenesis will completes inside the male reproductive structure, male gonad, that is called as the testis. So during the spermatogenesis process completes there in the seminiferous tubule of the testis. 2-step process is there: formation of spermatid and the spermiogenesis, formation of spermatid on the germinal epithelium, and spermiogenesis on the Sertoli cell. So formation of spermatid is a process that is being completed on the primordial germ cell or from the primordial germ cell to through the mitosis and the meiosis. 3-step process is there, that is multiplication phase. The primordial germ cell will undergo in the process of mitotic division. The repeated mitotic division will give rise the n number of the new cell, that is called as spermatogonia, that all are the diploid cell. Any one of the spermatogonia will enlarge due to the accumulation of nuclear and the cytoplasmic material, and will finally convert in form of the larger cell, that is called as the primary spermatocyte. So from conversion of spermatogonia to primary spermatocyte is the part of growth phase, because there is no division occurs here on. Now what happens, this primary spermatocyte is moving into the maturation phase, or we can say the complete developed spermatid is being produced through the meiotic division. So maturation phase is completely the phase of meiotic division. Here the primary spermatocyte will undergo in the two subsequent meiotic division and will form 4 haploid spermatids. From here the spermatids are moved on to the Sertoli cells, and they are just going to attach to get the nutrition and further development of them. And here that process of spermiogenesis will begin. The process of spermiogenesis is the metamorphosis only, where the spermatid is going to convert in form of the sperm. What happened during the process? The spermatid, which is heavier in the weight and non-motile, is being converted in form of the lighter structure and the motile structure, because it has to travel the long journey into the female reproductive tract. Then later on, what are the changes there? Spermatid nucleus is going to form the sperm head. The Golgi body of the spermatid is going to form the acrosomal membrane and the acrosome. The centriole is going to form the tail and the proximal and distal centriole. Axial filament is being produced from here. The middle piece is being produced from the mitochondria of the spermatid. Then we have discussed the structure of the sperm, 4 different parts. The head made of nucleus covered with the acrosomal membrane and the acrosome. Second one is neck with the presence of primary and second proximal and distal centriole. Then third one is called as the middle piece, formed of the mitochondria, provide energy for the movement of the sperm. Next one is called as the tail, final part made of the centriole only, help in the movement of the sperm, that is the longest part of the sperm. So this is what all about the structure of sperm, the spermatogenesis. In the next lecture we'll discuss the oogenesis. Till the time, thank you.